Oh, wait, you're listening.
Okay.
All right.
Okay.
All right.
You're
Listening
to Radio Lab.
Radio Lab.
From
WNYC.
See?
Yeah.
Oh, let me tell you, there is nothing like the sheer elation of discovery. And I was thinking, this is the end of malaria. This is the end of everything else mosquitoes spread. Wait a minute, ticks spread Lyme disease. We can probably get rid of that too. I thought,
So in the morning, you were like,
You're singing to the turtles in the park and
pretty much. And I give myself a full day of being who, and then I started thinking, but, what if something goes wrong?
I'm Jad Abumrad.
I'm Robert Krulwich.
This is Radio Lab. And the guy that you just heard is Kevin Esvelt. He is a scientist. He was talking to our producers, Soren Wheeler and Molly Webster.
About CRISPR, which is a technology. Actually, it's a gene editing technology that can reshape life, actually.
Yeah, and we ended up doing an entire show about this.
Mm-hmm.
Yeah, and we called it Antibodies Part 1.
I do remember that.
As if there was going to be a Part 2.
and that's like, name that. It's like telling someone you got them a birthday present but you haven't yet.
Yeah, it's true.
Maybe we should just own up. Radio Lab listeners, we did not get your birthday present.
Let's just that seems mean. No, we meant to. We meant to have a part 2, but we were doing a story, it fell apart.
Life doesn't always work out.
The tape sucked, frankly, and we thought it was going to be a story. It just turned out to be a story.
But now what we're going to do is we're going to pay you what you're due.
This is the part 2.
Yes, this is the part 2.
Finally, part 2, because CRISPR, in the time that we did the thing till now, has gone banana crazy.
So much has happened, really.
Yeah, like every day in the science section, which I know we all read religiously, there is a CRISPR thing.
Yeah.
So just to get us started, we're going to play you the original piece for those of you who never heard it, just to sort of set the baseline, and then we're going to come back and tell you all the stuff that has happened since.
Yeah, yes.
All right, so let me explain to you how I got started with this.
You were at some kind of an affair?
Yeah, so I'll tell you how it got. I was at a party. It was a conference where they had a lot of different people with different disciplines come together. One of those. There are panel discussions and various things. We were at one of the functions, and it was a situation where dinner hadn't yet been served, and there was a lot of booze being served. Everybody was drunk on an empty stomach. So I was standing there with some biologists.
Oh, they're the fun ones, the drunk biologists, yes.
As my people apparently, and they started to lose their shit, genuinely lose their shit about this thing called CRISPR. And I have never seen scientists this excited about anything. So I was like, what is this thing? What is CRISPR? And they were trying to explain it to me, but they couldn't slow down enough for me to get it. I gathered it had something to do with genetics, and then at one point, one of the biologists turned to me and he was like, I'll tell you what it is. I can use CRISPR to take a little dog and poof, make it into a big dog. Give me a Chihuahua, I could turn it into the size of a Great Dane. And I was like, no, you can't. He's like, yes, I can. I can do it with CRISPR. I was like, what the hell is this thing?
You want me to sit here as usual?
Yeah, if you sit here, I will get out. Fine.
No.
We'd be sitting here together.
So what happened was I came back and I immediately called science writer Carl Zimmer, because I just figured for this kind of thing, this is a Carl thing. I gotta talk to Carl. So I basically asked him, why all the fuss? Maybe it was just the alcohol, but maybe there's something really happening here.
Oh, there's something totally happening here. I mean, it's big.
You started at the beginning.
So you can actually find the first reference to CRISPR in a 1987 paper from some Japanese scientists. They basically described something weird in E. coli, and they said, we don't know what this is.
E. coli are bacteria inside humans, and like all living things, E. coli is made up of DNA, A's and T's and C's and G's, and what happened was that these scientists were reading a chunk of that genetic code when
they found this really strange stretch of DNA.
Strange how?
Well, so basically what it was 5 identical sequences in a row. And then they were separated by very short sequences in between them that were all different from each other.
These little blurps would be like...
And they looked at this and they're like, what? This is nothing like we've seen before.
Repeated sequences in bacterial genomes are kind of unusual.
Seems very strange.
Some biologists felt that, there must be a purpose for these.
Among those purpose seekers,
Jennifer Doudna, University of California, Berkeley.
she's a cell biologist.
Yeah,
So it's Doudna, not Dudna.
it's Doudna. I used to be called the dude sometimes in school, but
In the movie, she will be played by Jeff Bridges.
Right. Anyhow, as time goes on, scientists start seeing these little blurb repeats everywhere,
Yes.
or at least in bacteria.
Lots and lots and lots of species of bacteria. They say, okay, wait a minute.
that's kind of cool.
They're finding it so often that they decided they had to give it a name.
Is this where the name CRISPR comes from?
Yes. The full official name is
Clustered, regularly interspaced, short palindromic repeats.
I don't know why they called it CRISPR. It's kind of a.
It's like a furniture manufacturer.
It sounds like an app.
But now, scientists have this puzzle.
If nature at this level preserves something intact, here and here and here and here and here and here, and some of these here's are creatures that have been around for hundreds of millions of years, you figure, well, whatever this is,
It's doing
it's
something.
doing something.
But what?
It doesn't take very long before the first big clue comes up.
All right, fast forward. 2005. Now scientists have these big searchable databases of DNA sequences. So some scientists think, well, let's do a search. Let's see if these repeating patterns we keep finding match anything else that's out there in the world.
And these scientists are using computers to just line up these stretches of DNA with thousands upon thousands of different species, and then click. All of a sudden, they discover
That those bits of DNA between the repeats, the stuff in the middle, those blurps,
these are matching virus DNA. Like you can find viruses with genes where these little, these little.
do the bacteria have virus inside of them?
Yep.
Does that mean that a virus brought it into these cells? Does
Well-
it tell you anything about the origin of it?
The first recognition was this is virus DNA. Somehow, all these bacteria have little snippets of virus DNA wedged in these particular places in their genome.
Which is a little weird if you think about it. I mean, these are totally different creatures. It would be like inside a human finding a little bit of mosquito DNA.
Or how do we interpret this?
Well, actually, there was one scientist, his name's Eugene Koonin, who looked at these results and just said, okay, I get it. It's a defense system.
Why would he think that?
Because he's a brilliant man.
What do you mean? If I went to a large sanitation dump and I found a teeny bit of human hair, why would I think, oh, I get it, it's a defense mechanism? I wouldn't know. It's just like a bit of human.
Right. Well, you see, that metaphor might sort of betray your lack of skill in microbiology. I'm just saying, like, this is not a dump, all right? Bacteria are not going to just let virus DNA get into their genes willy-nilly, okay? Remember, viruses are the big enemy.
Right.
If you're bacteria, viruses make your life a nightmare. Think about in the ocean, okay? The ocean is full of viruses, and viruses kill up to 40% of all of those bacteria every day. Every day, yeah. And we know that they have defenses. What Eugene Koonin said was, okay, I'm going to bet that these bacteria are somehow grabbing pieces of DNA from viruses, and then they're storing it. And now they have a way of recognizing those viruses as they come in later.
It's like little Polaroid shots of the enemy.
Right.
Know thy enemy.
Yeah, like a most wanted poster.
What we call the mugshot,
This is Eugene Koonin.
leader of the Evolutionary Genomic Group at the National Center for Biotechnology Information.
He's the guy that Carl referenced who thunk up the whole idea that maybe these bits of virus DNA inside the bacteria is the bacteria trying to defend itself.
But really, if I would credit myself with anything here, it was not so much getting it, because when you see these identical sequences, that gets pretty obvious. It is figuring out how the mechanism was likely to work.
So can you walk us through how the mechanism is likely to work?
All right. what happens is,
You know, when a virus comes in to a cell, it just kind of explodes and kind of releases naked genes, basically.
If you're this bacteria, these things might take over your cells, so you've got to respond.
most of the time, you have multiple weapons of defense.
If you've never seen this virus before, usually the first thing you do, says Eugene, is you send out these enzymes to attack the viruses. They're sort of like the ground troops, and they fight really hard.
But much of the time, they fail, and then no one will hear about you again.
They're not terribly sophisticated fighters, so very often the virus takes over, the bacteria dies.
But there is some non-zero probability that you actually survive the attack.
If you do, then what the bacteria will do is send in some new enzymes to basically clean up, to go out, find any stray viruses.
And then cut the enemy DNA into suitable small pieces.
And here, he says, is where you get to the storage part. Those enzymes will then take those little bits of virus and shove them into the bacteria's own DNA, right in those little spaces between the repeats.
Right there and nowhere else.
So I use those spaces in my own DNA as a storage facility?
Yes, if you will, you use it as a memory device.
Because here's what happens. Next time that virus shows up, sprays its genes everywhere. Now you are prepared. And this is where the CRISPR story really gets going. Because instead of sending out the ground troops who are probably going to get their asses beat, now you can actually send out the big guns. And in fact, what the cell does is it will manufacture these special molecular assassins. And they'll give those assassins a copy of that little bit of virus DNA it has in storage, basically saying, here, take this mugshot. If you see anything that matches this pattern, kill it.
And these attackers, do we know what one of them looks like?
Yep. So we know what the protein looks like. It actually looks. I would describe it a little bit like a clamshell.
Sort of imagine Pac-Man, but kind of misshapen and rough. And each one of these guys,
What it has is a copy of that virus DNA
it's got the mugshot.
that it's kind of waving around. What then happens is that
Whenever the Pac-Man bumps into some virus DNA,
it pulls apart the DNA, unzips it,
reads it. If it's not the right one, it goes on. Nope. Mm-mm.
and if that RNA has the same sequence, then click, click, it just locks in.
And if that happens, then the DNA is trapped, and molecular blades come out.
And chop.
Cutting its head with a mighty blow.
Yeah.
Oh, so this is smart scissors.
So it's like, are you like the thing I got? Are you like the thing I got? You're like the thing I got. Snip, snip.
All right, now we're gonna kill.
Oh,
yeah.
I see.
And it has to be exact. It has to be an exact match.
When scientists first discovered this whole system, they were fascinated.
They were working it out. They're like, oh, okay, then this happens, and this happens, this happens. Cool.
But then, in walks
The dude.
Jennifer Doudna with a crazy idea. Well, I don't know if it's crazy, but radical.
This could be an amazing technology.
This is a tool.
This is a tool.
Yeah.
Right.
This is a tool that we can use to cut DNA where we want to cut DNA.
Her basic thought was, why don't we turn this defense into offense? Because these things, they seem to be really good at cutting, and yet they only seem to cut the things that are on their mugshot. So maybe I could just replace what's on their mugshot. So instead of them going after viruses, maybe they could go after a gene that causes Huntington's disease or hemophilia. For example, and this is actually something that's been done, say you got a mouse with something like hemophilia. This is a disease that's caused by one bad gene. So what you do is you take these little surgeons, you give them the mugshot for the bad gene, then you stick the surgeon with the new mugshot in a mouse,
Then you set it loose,
and just like it's programmed to, it will find that gene.
and click, click, chop. The scissors will end up cutting exactly the gene you wanted to cut.
So the bad gene's gone, Now the question is, how do you put in the good gene?
Right.
It turns out, actually, according to Jennifer Doudna, that that's actually not as hard as you would think. Yeah, apparently what you do is just throw this new good gene kind of in the neighborhood of where the old gene is, just in the general vicinity.
You don't have to get super precise. I mean, it turns out that there are repair enzymes that are probably continually surveying and checking for breaks.
She says what'll happen is that inside the cell, these repair crews will come along, they'll see the break, they'll see the good gene just sitting there next to the break, they'll be like, all right, I'll just stick it in.
Put the pretty guy in this space.
Exactly.
So we take advantage of a natural repair pathway that cells have.
They trick both the cutters and the fixers.
Yeah, now we're not assassinating anymore. Now we're actually engineering. We've gone from killing to refashioning.
Well, haven't we been designing, doing genetic, a form of genetic engineering for years?
Yes, but not like this.
Genome editing technologies have been around for a long time, but none of them have been as powerful as CRISPR is.
That's Beth Shapiro from UC Santa Cruz. She was actually one of the biologists that I drunkenly talked to at that thing.
Was it a modern art museum? I can't even really remember.
I don't remember either.
Must have been quite an evening to have the setting be so vague.
Anyhow, here's how she put it to us. Back in the day, this was just like 2 years ago, you would have these gene editor things. You would take one, put it in a cell.
And what happened before was you would give it some instructions about where to go, and it might go there, but it might go to somewhere that's kind of related to where that was.
So it's like, you take a right at Staten Island, but it takes a left.
And not only would it take a left at Staten Island and not find there, but it would have cost you a fortune and taken up 6 months of your time to get that thing. And now, it's really easy.
You just give it that mugshot.
And it goes, I'm going to find that guy. Exactly.
So it seems to be pretty precise, and it's cheap. Like the old tools would set you back about $5 grand just to use them once. CRISPR, about $75. And here's the kicker, says Carl. It seems at the moment that you can take these things out of bacteria, stick them into almost any other creature, and it still works.
You can use the same CRISPR system on anything.
Can you do it if corn is vulnerable
Do it in
to
corn.
a certain pest? You can do it in
Do it
corn.
in corn, do it in anything. I'm waiting for someone to say CRISPR doesn't work in species X, and I have not heard of that.
So basically what you have for the first time in science is this gene editing technology that is cheap, precise, and possibly universal. And Jennifer Doudna says the moment the full impact of that landed on her,
I literally had, the hairs on the back of my neck were standing up.